Then we come to the environmental field. So it's not like we can do only it in animals, humans, or plant. We can do it in environment. The best example is, you can say, during COVID, you might have heard a lot of story about collecting the data from sewage line or the flowing water in the community, and try to find out if that community anyone has got COVID. Once you get the positive sample, then door to door surveillance started for that community. That is one very recent example, that's why I'm giving you. But there could be a lot of things, like I said, the soil quality. Or yesterday someone was saying that they got some plastic degrading bacteria. Again, these will be found in the place where there is lot of plastic, lot of garbage, lot of, let's say, industrial waste. So if industrial waste is just dumped in the ground, but still when you take the soil sample at different height of that place, you will find out lot of bacteria, fungi, or algae growing in that thing. So what are the things which make those bacteria or the microbiomes in collective, I will say, to survive in that kind of conditions? That is one thing you can do. So there are a lot of things. I am showing it here. You can see that you can do a lot of things, like quorum sensing is again how these microbiome, the microbes interact with each other. You can do DNA binding domain study. You can do transcriptomics of the bacteria, either single or in population. You can find out the regulatory element present in that kind of things. You can make, so IGEM is a competition which take place every year, where there are a lot of examples where people have used biosensors. They created synthetic biological biosensor system. They did metabolic engineering to increase one kind of, let's say, bioethanol. They want to increase ethanol production using plant. They will change the path. The researchers change the path of something in, let's say, bacteria or in plant, and that is called kind of metabolic engineering, and they can increase the yield of that thing. Again, genome editing, I just told you, in plant you can do that, in bacteria also. So the bioremediation is one thing which is coming up very fast. The pace is picking up because we have a lot of disposals, and we need some method, maybe biological or anything, to just get rid of that garbage, so, or get rid of the toxic elements. So that is another field people can look for. So this is a unique one, is called NGS in, I will say, food industry. So in food industry also, before packing any sample of a food, before delivering the sample to a customer, the what people do, quality checkers, they do is they just get a hand on random samples from a lot. They go and they check for bacteria like salmonella, which is very common nowadays, or other kind of pathogens. If other kind of microbes or salmonella or other things are present in samples of the food, the whole lot is burnt, lot is discarded. So if anyone follows CDC come on regular basis, come with a lot number and food, kind of food where we should discard that food, and they found salmonella and other things. They keep on advertising it very frequently. The last thing uh, I think it was onion or peanut butter, where CDC said that they found traces of salmonella in that, and they gave the lot number. If anyone is using the uh, product from that lot number, please discard it. So this can be used. Or some people are allergic to peanuts, or allergic to some other kind of foods, gluten allergy or something. So that also, so we have already have allergen library database present, and we are coming up with lot of more allergen. You can just go, you can just help in that also. You can, in food industry also, there is a lot of scope in NGS. So these all kind of things NGS is used. There are a lot other things, but you people will know, you will, when you will follow the NGS future trends. So this is just for example, I showed you that transforms some different. I try to cover different aspect of that.